Okay, so this is a summary, and maybe it'll help you to visualize everything that's happening in cell respiration. Keep in mind that we're not super focused on the numbers, like the number of NADH and FADH2 that's made. Not all of that stuff is super important. It's more important that you know the general processes, where they happen, and where a majority of the ATP are made, where a majority of the CO2 are made, you know, how ATP are made in each step, etc. So we're going to go through that right now. So our first step in cell respiration, what I've drawn here at the top is glucose. And I'm technically only drawing the carbons from the glucose. I haven't actually drawn the oxygens and the hydrogens. But remember that glucose also has oxygen and hydrogen. It's not just carbon. So in our first step, this is going to be called glycolysis. This is our very first step. Glycolysis. It literally means glycolysis, the breaking of sugar. Lysis means to break. And the location of this is in the cytoplasm. And it's very important, things you could be asked on a test. You should know that this process happens whether oxygen is present or not. It happens universally in every living cell is running glycolysis. It doesn't depend on oxygen, it doesn't depend on mitochondria. And in essence, all that really happens in glycolysis is that one glucose goes in, and after a whole bunch of steps, two molecules called pyruvates come out. And a pyruvate is a 3-carbon sugar. So we went from a 6-carbon sugar to a 3-carbon sugar. Notice that no carbon dioxide is made in this step. What is made in this step, now this step has a cost to it. It costs 2 ATP to run glycolysis. They go in, but it makes 4 ATP. A test question would probably be worded, how many net ATP are made in glycolysis? Or how many ATP are gained in glycolysis, meaning, you know, above and beyond what it costs. So our net is 2. The other thing that's made in glycolysis is a coenzyme called NADH. Now, it started as NAD+, and it gets turned into, technically it's 2 of them that are made, and it turns into a reduced form NADH. And that's going to be important later because those hydrogens, when they're transferred, hopefully to oxygen in the end, are going to generate energy, ATP. Now, so once we've made our pyruvates, you've got 2 options. And this is where it matters whether oxygen is present or not. Even though the oxygen is not going to actually be necessary yet in the process, if there's no oxygen, we're not going to go any further in the process. The next thing that's going to happen is going to be called fermentation. And the products of fermentation depend on the organism. If it's a bacteria, it's usually ethanol. Technically ethanol and carbon dioxide. And that's what makes bread rise, and that's what's used to make beer and wine, etc. If it's a human or not an animal, usually the product is lactic acid. So it is not true that we can't make ATP if there's no oxygen. We can. But we can't make very many because we would only be able to run anaerobic respiration, which means we would run glycolysis and fermentation. We would also call that the fermentation pathway. It doesn't make enough ATP to keep us alive. And lactic acid is sort of a toxic waste product. The acid lowers the pH, and that's what causes the burning in your muscles. And so you can't rely on it for very long. But the purpose of fermentation, and this is really important, kind of a little asterisk here, is that it oxidizes NADH back to NAD again. Why? Who cares? Because NAD was necessary to run glycolysis, that coenzyme. It's sort of like, and I gave the analogy, if you are bringing a bunch of groceries into your house and your hands are full, you have to put down the groceries you just brought in so you can go out and get the rest. So NAD picks up hydrogens from glucose during glycolysis. At the end of glycolysis, it's got to get rid of those hydrogens because you have a limited amount of NAD. So it's got to go back to being its oxidized form, and then glycolysis can keep going. All right, if oxygen is present, then everything else that's going to happen is going to happen in the mitochondria. And so that's why I drew this little line here, because everything else is now going to happen in the mitochondria. So now we've entered the mitochondria. The next process is going to be the citric acid cycle, or it's also called the Krebs cycle. Don't mix it up with the Calvin cycle from photosynthesis. So Krebs or citric acid cycle, and its location is the matrix of the mitochondria. And in a mitochondria, just a reminder, the matrix is this part, the middle. All right, now, there's a little step while the pyruvate is entering the mitochondria that I have drawn out here, and it's called oxidation of pyruvate. And I told you it's not really considered a major part. The three major parts are glycolysis, the Krebs cycle, and the electron transport chain. But I have to kind of mention oxidation of pyruvate because it explains where the CO2 goes, or where those carbons go. So oxidation of pyruvate is where our pyruvate loses one of its carbons, and that becomes CO2. And this is also the O is coming from oxygen. And that pyruvate gets turned into a new molecule called acetyl coenzyme A. And so you make two of those because you had two pyruvates. You now have two acetyl coenzyme A's, or it's written out as acetyl-CoA. And notice this is a 2-carbon molecule. So this occurs as pyruvate goes into the mitochondria. It gets converted from a 3-carbon molecule to a 2-carbon molecule, and that third carbon has now been released as carbon dioxide. The other 2 carbons that are in acetyl coenzyme A are also going to be released during the Krebs cycle as CO2s. So each of these carbons is going to become a CO2. And by the end of the Krebs cycle, and this is super important, by the end of the Krebs cycle, all carbon and all oxygen from the glucose are now going to be converted into CO2. And that's released. So a majority of your CO2, for test purposes, are made here in this second step, the Krebs cycle. Now, what else is made in the Krebs cycle? Well, you also make, each turn of the Krebs cycle makes an ATP. So you make a total in the Krebs cycle of 2 ATP. Remember, we made 2 in glycolysis originally. So if I highlight these, we made 2 ATP here in glycolysis. I know this is getting very messy, but hopefully you're following me here. And we made 2 ATP total here in the Krebs cycle. So we've only made 4, not very much. But the most important thing that the Krebs cycle does is it makes a whole bunch more NADH and a whole bunch of FADH2. And I'm not even going to put numbers because I really don't want you to memorize how many. You should know the ATP, but NAD and FAD, you don't have to know how many. In fact, if I really wanted to be technical, you also make some NADH here in the oxidation of pyruvate. All right, so now comes our very last step. That is going to be the electron transport chain. Now, the electron transport chain is the only step that's going to make ATP in that special way called oxidative phosphorylation. The other ones that have been made up to this point were made by substrate level phosphorylation. And a reminder of the difference between them. Substrate level makes them one at a time, like the farmer picking corn on his farm one at a time. Oxidative phosphorylation is going to make a whole bunch of ATP at once, like a farmer that has a machine that can harvest a whole bunch of corn at one time. And this is going to happen in the cristae, or the inner membrane of the mitochondria. Now, the only part of glucose that goes into the electron transport chain is the hydrogen. The hydrogen is the most important part of the glucose as far as making ATP. Because what's going to happen is all the carbon dioxide is gone. So the carbons and the oxygens are gone. All the NADHs are going to get rid of their hydrogen and go back to being NAD. The FADH2s are going to get rid of their hydrogens and go back to being FAD. The hydrogens are going to split into 2 things, and I'm not going to get into all of it now, but they become a proton and they become an electron. In other words, the electron is broken off the hydrogen. The electrons go down the electron transport chain. The hydrogens are going to get pumped into the intermembrane space. And again, refer to my other lecture if you want the details on this, but they're getting pumped through into this space here. And then what actually makes the ATP, if you're asked what part of the electron transport chain is where the ATP are actually generated, that's going to be ATP synthase, the enzyme. The hydrogens that built up out here are going to flow back through ATP synthase. So it's the flow of the hydrogen ions, which are technically protons. And this is going to generate an additional 32 ATP. So in the end, we make about 36 ATP, but 32 of those 36 are made here in the electron transport chain. Almost all your ATP are made there. So Where are the most ATP made in cell respiration? The electron transport chain. How are the most ATP made? By oxidative phosphorylation. Where does the electron transport chain happen? In the cristae. What actually travels down the electron transport chain proteins? Electrons. But what generates the ATP is the hydrogen ions, the protons, flowing through that enzyme, ATP synthase. So that is sort of a summary of everything that happens. Oh, super important, I almost forgot. This is where the oxygen finally comes in. O2 is needed here. Oxygen is what's sort of sucking all the electrons down the electron transport chain. It's extremely electronegative. And so by sucking the electrons down the electron transport chain, pulling the electrons towards it, that's what generates the energy to pump the hydrogens. So if I go back, if I actually just create a new slide really quick, here's your giant mitochondria. Here is your cristae. The hydrogens, the electrons from NADH and FADH2 are going down the electron transport chain. So here's my electrons, and they're getting passed from protein to protein. This is the electron. That, because those electrons are getting sucked towards oxygen, waiting here at the end, pulling the electrons towards it, that they're moving towards something more electronegative, and that allows this energy to pump the hydrogens out here. So the hydrogens all build up. They now flow back in through our enzyme, ATP synthase. And this is where the energy to make ATP is going to really happen. The hydrogens flow back through ATP synthase, and everything hooks up, the hydrogens and the electrons, to form water. So all your water is made here in the last step. And the only place you actually need the free oxygen is here in the last step. Oxygen becomes water in the very last step. And that is where you're getting all the energy to make all those ATP. So to say it one more time, NADH and FADH2 give up their hydrogens. The hydrogen splits into two things. A hydrogen ion, which is a proton, and the electron that comes off of it. That's why there's a positive charge here. It's missing an electron. The electron goes down an electron transport chain. What pulls it down the chain? Why does it want to go down that chain? Because oxygen is waiting at the end, calling to it. This movement generates energy because the electrons are moving towards something more electronegative. And whenever they do that, it generates some energy. The energy pumps the hydrogens out here to the intramembrane space. And then the ATP are made when the hydrogens flow back through, from high to low, through this enzyme, ATP synthase. The hydrogens and the oxygens hook together and make our final product of water, and we make an additional 32 ATP. So our total ATP is about 36. 32 are made in the electron transport chain, 2 were made in glycolysis, and 2 were made in the Krebs cycle. These were made substrate level, that method that's not very effective, and these 32 were made by oxidative or chemiosmotic phosphorylation, which basically refers to the fact that it has to do with an electron flow and this proton gradient, that there's an electrochemical gradient there. So hopefully that summarizes the steps in cell respiration and gives you maybe a little bit more of a visual on it. Again, go back to the regular lecture if you want more details, and also come see me if you have any questions.